We will switch gears a little bit now, and I'd like to introduce Dr. Terry and Dr. Kay from Silent Spring. I will let them share their slides, and they're going to talk to us about environmental risk factors. So welcome. Thank you for joining us.
Thank you very much for having us. Wonderful. So the two of us this morning will give an overview of breast cancer incidence trends in the world. I'll do that part. And making a case for why we can't ignore research and study around environmental chemicals. And my colleague, Dr. Kay, will be making that argument. So as you've heard, early onset cancers are going up. On the left-hand side, you'll see cancers in 18 to under 50-year-olds and females. And on the right-hand side, you'll see males. And of course, breast cancer rates are the highest of any of these cancer types. Thyroid, which is in orange, is under breast cancer. But what you really see very quickly is that even though cancer rates are going up in a number of different cancers in females and males, 2/3 of all cancers diagnosed under 50, this is true globally, and it's true in the US, are diagnosed in women. So there still is a much larger burden in women under 50. So, and globally, you can see the pattern of cancers has changed dramatically over the last 30 years. Countries like India, which used to have cervical cancer higher rates than breast cancer, now has breast cancer rates higher than cervical cancer. So globally, you can see breast cancer rates have been sweeping the globe. And as I mentioned, 2/3 of these early onset cancers are in women. So this is really one of the largest health disparities we know of in cancer. So the dots here represent, each dot is a country. There's 185 countries in this graph. And what you can see over time over the last 30 years is there's definitely this shift from the right to the upper left quadrant. On the y-axis is breast cancer incident rates, and on the x-axis are fertility rates. So yes, it is true that fertility rates have been going down over time. And yes, it is true that breast cancer rates are going up. And those two things are correlated. But what we want to make a case for is that that cannot solely explain what's going on, particularly for women under 50, which is what this graph represents. And in fact, what you can see is for a similar range of fertility, you can see very large differences in breast cancer rates. And that would be represented, I think, sorry, the rectangle's a little off here, but that would be represented just in the far left, where you see similar fertility, but almost a 7, 8, 9-fold difference in breast cancer rates. Similarly, on the rectangle on the bottom, you see very dramatic differences in overall fertility rates across these countries. But still very similar breast cancer rates. So all of this is to say that there's quite a lot of variation, and that fertility alone cannot explain why these breast cancer rates are soaring around the world. And then just to represent certain countries, actually, one major trend over the last 30 years is 30 years ago, breast cancer in the US used to have the highest rates in the world. And now you can see we're kind of in the middle of the pack for early onset cancers. So what's important and what we've learned over the last 20 years in particularly is that the breast is most susceptible to carcinogen during certain windows of susceptibility, and we call those WOS for short. And that's really when the breast tissue is changing in form and function. So that would include in the fetal and early infancy, the pubertal window, during pregnancy and lactation, and the involution that happens postpartum in the breast tissue, as well as you just we heard an excellent talk about the menopausal period, which is a much longer period than these other periods, but also when the breast is really changing in form and function. The other thing we know now from the last 20 years of science is that the somatic mutations, so the mutations we get after we're born, that are based on both exogenous environmental factors and then endogenous factors that just change when cells divide, that most of those mutations occur much earlier in life. And in the top right quadrant, what you can see is a schematic of how cancer and aging kind of mirror each other, because cancer is a disease of aging. But what you can see with the brown dots are those are somatic mutations. So not what we're born with, but most cancers occur because of somatic mutations, not germline mutations. And those actually, 50% of them have been estimated to occur before the age 25. So now I'm going to pass it to my colleague, Dr. Kay, who's doing really fundamental work in understanding then how these chemicals may affect breast cancer risk.
Thanks, Mary Beth. So that was a great introduction. I'm now going to tell you some about what we do at Silent Spring to understand environmental exposures that may be underlying the high rates of breast cancer in the US and worldwide. So at Silent Spring, we follow a cancer prevention science model because, of course, most women would prefer not to get breast cancer in the first place, although treatment is also very important for people who do have breast cancer. So at Silent Spring, we have two main research focus areas. One is to understand the biological mechanisms that govern breast cancer development and how chemical exposures are involved in dysregulating those processes. And then our other research focus is to understand where and how people are exposed to these chemicals. Because with the combined knowledge about how people are exposed and what those exposures do, we can take actions to protect people. And this can include things like educating the public about potential exposures, supporting regulation of hazardous chemicals, and encouraging reformulation of products that use those chemicals. So I'll take you through just a couple things that we do at Silent Spring to approach these topics, starting with the biology of breast cancer. Now, there are different ways of studying the biology of chemicals that increase breast cancer risk. Epidemiology studies attempt to link health outcomes like cancer in humans with chemical exposures. But these studies are limited in their ability to tie specific exposures with specific outcomes because people have such variation in their genetic susceptibilities, and we're all exposed to hundreds of chemicals at various life stages. So it's hard to reconstruct the lifetime history of relevant exposures, particularly when we're trying to focus on things that happened during those critical windows of susceptibility that Mary Beth talked about in the early ages, such as, prenatal development, puberty, and then menopause. And plus, since cancer takes a long time to develop and it's influenced by diverse biological mechanisms, it can be hard to identify specific exposures that likely contributed to this long latency disease. So after human studies, people generally find studies in animals to be the next strongest evidence for chemicals to have certain health effects, because mammals often have similar responses to chemical exposures as humans do. And you can be more specific about exactly which biological changes the chemical is causing. Of course, animals are not humans, so there is uncertainty, and also animal studies are pretty expensive. So cell-based testing is also a good way to identify specific effects of chemicals because they're tightly controlled. You can use cells that are derived from human samples, and they're faster than human and rodent studies, so you can test a lot more chemicals. But these studies are also somewhat limited because you lose all of the interconnected complexities of a whole organism. So recently we published a paper where we compiled a list of chemicals that have evidence they could increase breast cancer risk. And we started by using databases from the International Agency for Research on Cancer and the US government to identify nearly 280 chemicals that induced mammary tumors in rodents. But since these studies are so time-consuming and expensive, not that many chemicals have been tested this way. So we stepped back and asked ourselves about how else we can find potential breast cancer risk factors based on what they do in cell-based assays. So we approached this by thinking about what we know about processes that increase breast cancer risk. So, of course, one thing that chemicals can do to cause many types of cancer is to damage DNA and cause mutations, and this is true for breast cancer too. But breast cancer is also influenced by hormonal activity, as we've discussed in this forum today. So increased estrogen and progesterone are known to increase breast cancer risk, as is clear from epidemiological studies showing that older forms of hormone replacement therapy that used combined estrogen and progesterone treatments increased breast cancer risk among postmenopausal women. Of course, newer formulations pose lower risks, but these earlier studies showed that this combination of estrogen and progesterone could increase breast cancer risk. And that is because these hormones cause breast cells to divide, and too much hormone signaling can overstimulate cell division. And this uncontrolled cell division is one of the defining characteristics of cancer. So one way that a chemical can increase hormone signaling is by activating the hormone receptors directly. So we know that estrogen receptor activation is important for breast cancer because many breast cancer treatments block the estrogen receptor, like tamoxifen. Other breast cancer treatments work by blocking hormone production. So chemicals that cause cells to produce more hormones can likely increase breast cancer risk. Now, in doing this study, we were able to use publicly available data from EPA's screening chemicals for their ability to activate the estrogen receptor and increase production of estrogen and progesterone. Unfortunately, there are no good screens for chemicals activating the progesterone receptor, so we couldn't include that in this study. But from EPA's chemical screening program, where they've tested nearly 2,000 chemicals for their effects on hormone signaling, we identified over 515 chemicals that were able to increase estrogen or progesterone production, and 267 chemicals that could activate the estrogen receptor, leading to a total of 920 chemicals plus ionizing radiation that we consider to be breast cancer relevant exposures. And many of these chemicals have more than one of these activities. Now, since DNA damage is another important way chemicals can cause cancer, we also wanted to characterize these breast cancer relevant chemicals according to their ability to damage DNA. So we used more US and international databases to identify chemicals that could induce DNA damage in animal and cell studies. A couple highlights of chemicals on our list include chemicals that have all four of these activities. These include certain types of dyes, as well as the drug diethylstilbestrol, which was discontinued actually because it was found to increase breast cancer risk among other cancers. We also have some chemicals that could increase hormone production, damage DNA, and cause mammary tumors. These include certain chemicals found in smoke, various pesticides, and some other types of dyes. And of course, we have some well-known endocrine disruptors like phthalates, bisphenols, and parabens. So where are these chemicals found in our environment? Well, we know that rodent mammary carcinogens are in things like plastics, including styrene, polyvinyl chloride, bisphenols, and phthalates. Breast carcinogens are also found in auto exhaust and air pollution. They include certain pesticides, various types of solvents, flame retardants that may be added to furniture and home furnishings, as well as water disinfection byproducts. We also know that hormone disruptors that haven't necessarily been shown to cause mammary tumors are found in food packaging, personal care products, fragrances, chemical sunscreens, and various household cleaners. So once we know about what chemicals we should be concerned about and where they are, the next step is to figure out how we can reduce our exposures. So Silent Spring scientists did a study where we collected urine samples from over 700 participants and analyzed them for some common household product chemicals, including parabens, bisphenols, antimicrobials, UV blockers, flame retardants, and chlorinated phenols. And we found that everyone had at least 2 of these 10 chemicals in their bodies, and most people had 7 or more. So we then wanted to figure out how levels of chemicals in people compared to their use of personal care products, because this could reveal ways to reduce the levels of chemicals in our bodies. So first, we found that people who used more personal care products, including things like makeup, lotion, soap, and shampoo, had higher levels of chemicals in their bodies. So for example, everyone had detectable levels of a chemical called methylparaben, but people who used more products had higher levels. So simplifying your beauty routine is one way to reduce chemicals in your body. And I just want to point out that the x-axis here is showing the quartiles of how many products people used, not the number of products. So this 4th bar represents people who were in the top 25% of product users, not people using 4 or more products. So in this study, we also found that people who avoided products that listed parabens in the ingredients label also had lower levels of parabens in their bodies, even if they used a lot of products. So reading product labels and avoiding ingredients is another effective way of reducing chemical exposures. And I'll give you one resource or a few resources where you can help identify ingredients that you might want to avoid in products that you're considering buying. In a different study, we looked at how diet influences chemical body burden. So we had participants eat a diet consisting entirely of fresh, unpackaged food for 3 days, and we measured the levels of hormone disruptors in their urine before, during, and after their fresh food diet. And we found that eating fresh, unpackaged food could reduce levels of bisphenol A or BPA, as well as phthalates, which are endocrine disruptors used in food packaging. And the levels of these chemicals could be reduced in just 3 days of eating a fresh food diet. So now that I've given you a taste of how we identify potential breast cancer hazards and how we're being exposed, I'll show you a few free tools that can help you avoid those chemicals in your everyday life. But I also want to recognize that it's unfair for the burden of protecting public health to fall on individuals. And the problem of toxic chemicals in our environment isn't something that we can shop our way out of. At Silent Spring, we actively share our research with policymakers so they can set regulations based on the best available science and protect public health at the institutional level. So, that being said, what can you do to identify chemicals that you may be exposed to in your life and avoid them? Well, Silent Spring makes a free app called Detox Me, which is available online and on all mobile device platforms. And Detox Me gives tips to reduce exposures from different aspects of your life, like at home, in cleaning products, and in personal care products. The app contains over 280 tips that you can scroll through one by one. And we also have top 10 lists by categories, along with buying guides to make it easier to reduce potentially toxic exposures in your life. Another great tool that is made by a different organization that Silent Spring partners with is called Clearia. And this is a tool that you can use as a web extension or as a mobile app on your phone. And this provides consumers with information about what's in specific products so they can avoid harmful ingredients. So this app actually can interpret your ingredients list on a product that you're considering buying and tell you which of those ingredients might be harmful. Another app that sort of does a similar thing is called Yuka, Y-U-K-A. I really like this app because it can read the ingredients list based on scanning the barcode on the product. and also tell you about potentially toxic chemicals in that product. I know that some people here are interested in finding recommendations for non-toxic products, so these are some great resources to help you evaluate the things you're considering buying. So I'm going to wrap up there. Mary Beth and I will now be happy to take any questions that you might have.
Thank you very much. Very informative. There is one question in the chat. Thoughts about relative concerns with products for a woman's period, so tampons versus pads versus period underwear.
Yeah, so certain types of menstrual products have been shown to use certain potentially toxic chemicals. I think the type of product that is least likely to contain chemicals of concern are medical silicone grade menstrual cups. There are also certain types of other period products that are sort of organic cotton tampons. So, a product saying that it's non-toxic does not necessarily mean that it doesn't contain any potentially harmful ingredients, but those can be, indicators that the product contains fewer potentially toxic ingredients. And the apps that I showed you can help find the types of period products that are least likely to contain chemicals of concern.
And what was the name of the last app that you mentioned? I don't think you had a slide for it, but you called it out. They want to hear that name again.
Yeah, it's called Yuka, Yuka. It's also a free mobile app.
And one of the things that our patients are often having is MRIs. They're at increased risk for developing breast cancer, and some women do qualify for MRI screening. And there have been some concerns about the contrast dye that's used for MRIs, the gadolinium. Have you done any studies on gadolinium and risk?
Silent Spring hasn't done any studies on gadolinium in particular, and I'm not really familiar enough with the science behind that to really comment on what risk it may carry.
Okay. Well, I think, I'll just chime in there. I don't think that we know of any connection between gadolinium and breast cancer risk. And certainly we do find MRI screening helpful in some of our patients with elevated risk. So I wouldn't, if your doctor's recommending that you have an MRI, I wouldn't not get your MRI concerned because of concerns about the gadolinium causing breast cancer. There, you talked about cancers being driven by estrogen and progesterone, and we certainly know that ER and PR positive breast cancer is the most common type of breast cancer, but we also know the subtypes of HER2 positive breast cancer and triple negative breast cancer, and certainly triple negative breast cancer seems to be more common in young women. Any particular studies that you've done or comments that you might make about the different risk factors for the different types of breast cancer?
Sure. If it's okay, very quickly, I'll share my screen. I saw that question a little earlier, so it was a great question, and I do want to emphasize that knowing about the molecular subtype is extremely important for determining treatment status, and we are finding different types of risk factors based on some of the molecular subtypes, but for young women, so this is a study that we did a few years ago for young women, and you can see based on the different colors of the dots, for young women, luminal A, which is estrogen receptor positive cancer, is still the most common cancer in young women. So if you look at the y-axis here, this tells you the number, and you can see as one ages, the cancer rates get much higher, right? So luminal A is still the most common cancer, even in young women, and it's actually going up in young women. But it's much higher in non-Hispanic women than Hispanic women. And then you can see luminal B cancers, which are cancers then that are progesterone negative. And you can see that those cancers are also going up in young women across the board. But you had asked specifically about ERB2 and triple negative. So if we look here, yes, it's true that young women do have triple negative cancers and ERB2 cancers. But of the two, over the last decade, it's really just been the ERB2 cancers that have gone up. You can see down below the triple negatives have been more stable. But as you go across, because these the y-axis keeps increasing, you can still see that the bulk of triple negative cancer is going to be diagnosed a little later. Although, of course, you do see it in young women, particularly in non-Hispanic Black women across the board at all ages. It's double the rate. And so what's driving that is really a topic of a lot of research, including endocrine disrupting chemicals that Dr. K explained. There's some it's very early days for research, but looking in the lab to see if these endocrine disrupting chemicals can change a luminal cell into a basal cell. So a basal cell is what you're going to see in triple negative. So it's a great question. Thank you for asking. And we'll provide our email. So feel free to email us afterwards if you have more questions.